In 1984, the discovery of the human immunodeficiency virus was announced. At the time, U.S. Health Secretary Margaret Heckler thought that a vaccine against the newly discovered virus would not be too difficult to develop. She estimated approximately 2 years. Now, more than 30 years later, there is still no vaccine, which is the only strategy that had ever led to elimination of a viral disease. A vaccine trains the body to fight off a specific invader, known as a pathogen, and it teaches the immune system how to respond in the future. An ideal vaccine should be cheap to produce, easy to transport and administer without special equipment, and stable at room temperature. HIV is a novel and difficult pathogen and occurs in several subtypes, which are highly variable. Therefore, a broadly protective vaccine that covers all these HIV subtypes is nearly impossible. Soon after infection, HIV, which is a retrovirus, inserts its genetic material into human cells, where it remains completely hidden from the immune system within lymphocytes. As a result, any vaccine has to be able to repel all HIV attempts to infect and attach to host cells.
When the cell encounters the initial infection, the specific immune response gets wiped out. The virus is able to integrate into the host cell's chromosome and establish a permanent reservoir of infected cells. 4-NCO is an HIV envelope glycoprotein that attaches HIV to the cells that it infects and powers fusion of the virus with the cell membrane. This immersion makes it difficult for the vaccine to reach the virus. The HIV vaccine has been broken down into 3 generations. The first consists of vaccines that use a simple viral protein to elicit an antibody response, while the second produces a cellular immune response and stimulates anti-HIV CD8 cells. The third includes a CD8 stimulation component, which also initiates neutralizing antibodies. CD8 cellular vaccines do not block infection, but rather reduce the viral load in chronic infection. It can be speculated that the third generation vaccine would be the most successful as a preventative vaccine, as it consists of both humoral and cellular components. The humoral reaction is found in the stimulation of the neutralizing antibodies, which prevent viral particles from infecting new cells. As in a typical cellular response, the response is paired with CD8 stimulation, causing specific CD8 plus cytotoxic T lymphocytes to mobilize, which target and kill cells that express the antigens.
There are 2 types of general vaccines, preventative and therapeutic. A preventative vaccine is the most ideal form as it is essentially a cure. On the other hand, a therapeutic vaccine only delays illness in infected people. A vaccine mimics the shape and structure of HIV proteins, causing the immune system to produce antibodies and lymphocyte responses that recognize and attack HIV. The immune system can be educated to respond quickly to infection. The body's natural immunity prepared by the vaccine is hoped to be effective in fighting off real exposure. There have only been 4 large phase 3 efficacy trials of HIV vaccines in humans. The first 2 were differing versions of a vaccine called AIDSvax, but it was found that neither vaccine prevented infection. The third one, the STEP trial, looked promising in animal studies but had no effect in humans. The last trial involved a vaccination called RV144 that reduced infection by 31% and revitalized interest in the search for a better vaccine. Keep in mind that while there is no cure, treatment is still available, and it is key that you get tested, especially if you're between the ages of 15 and 65 years old.